Good afternoon, everyone. Welcome to our MHRC seminar this afternoon, featuring Dr. Atsushi Asakura from the University of Minnesota Medical School in Minneapolis, Minnesota. He is in the Department of Neurology as a professor. Dr. Asakura has been there 17 years. Of course, before that, he got his PhD at the University of Tokyo in molecular biology. His first postdoctoral stint was at the Fred Hutchinson Cancer Center at the University of Washington in Seattle. He spent 4 years there. And then he moved to Canada and worked with Dr. Michael Rudnicki, a combination of McMaster University and Ottawa Health Research Institute. He spent 5 years there before being hired at the University of Minnesota. So his interests, of course, are in stem cells and muscular dystrophy therapy. And his title today is Muscle Stem Cell Niche and Reprogramming. So I'd like to welcome Dr. Asakura to turn on his video and his microphone and share a screen with us and begin our seminar.
Okay, just a second. So sharing, wait. Just a second. Sorry, I'm looking for the place. Yeah, sorry, I'm looking for the sharing place.
Sharing thing.
I mean, the
Down
icon,
below.
icon is gone. Oh, okay, I found it. Okay, I found it.
There we go.
Okay.
That looks very good.
Okay, thank you. Okay, just a second. Pointer, laser pointer. Okay, so thank you for the kind introduction, David. And actually, the Anthony scheme, he's of course faculties in the York universities, and myself were overlapped for 5 years in Michael Rudnicki lab when we were postdoc. So
Dr. Asakura, maybe change the way your slides are appearing. Show them as a big slide on the screen. You've got multiple slides showing up here. Maybe
right now, okay.
Yeah, put the muscle stem cell niche title slide as a full screen slide. Yep.
So you saw something multiple slides here, or?
We see your row of slides down at the bottom, the whole row of your slides.
Oh, okay, I see. Okay, just a second.
If you go to the top left and click on swap displays, you can change it. Top left, I think, maybe.
How about this? Still?
Yes, that's great.
This
Perfect.
is better. Okay, perfect. Okay. Anyway, yeah. So okay. So So Anthony and myself were overlapped for 5 years in Michael Rudnicki lab when we were postdoc, and actually we were in McMaster universities. when we were McMasters, I lived in Toronto, so I really want to revisit Toronto after COVID-19. Okay, anyway, let's start. So I belong to the Stem Cell Institute and also Muscular Dystrophy Center, where there are more than 30 skeletal muscle research groups. And so this place is one of the biggest muscle research centers in US. Okay, so as you may know that skeletal muscle is a highly regenerative tissues. So the cardiotoxin injection can induce muscle injury, followed by muscle regeneration. So that 4 weeks after muscle injury, muscle can be almost completely healed. So because muscle contains stem cell population termed muscle satellite cells, which are committed myogenic stem cells of adult skeletal muscle, responsible for the growth and regeneration in adult skeletal muscle. And they are located beneath the basal lamina, closely juxtaposed to muscle fibers. Okay, so the satellite cells are normally mitotically quiescent, but activated by injury or exercise. Activated satellite cells enter a cell cycle, and after several rounds of cell division, they stop cell cycle and fuse each other to make a multi-nucleated myotubes. And the minor population of activated satellite cells go back to quiescent satellite cells as a self-renewal. In our laboratories, we are working on muscle stem cells and therapeutic application for Duchenne muscular dystrophies, utilizing MDX mice. Today, I'm going to talk about vascular niche for satellite cells, vascular therapy for muscular dystrophy, endothelial cell signature in satellite cells, and circadian regeneration of muscle regeneration, regulation of the muscle regeneration. Okay, so the first topic is vascular niche for satellite cells self-renewal, which project is done by our talented MD PhD student, Mayank Verma, and postdoc, Yuko Shimizu Motohashi. And satellite cell self-renewal and maintenance are regulated by signals from surrounding niche cells, including muscle fibers, capillaries, and also fibroblast. It is proposed that disruption of the satellite cell maintenance is a trigger of muscle aging and dystrophy. Last decade, we have been studying on vascular niche for satellite cell self-renewal and maintenance. Recent work demonstrates the close relationship between endothelial cells and satellite cells. So around 80% of satellite cells are co-localized with the capillaries, like this in skeletal muscle, suggesting that proximity of capillaries is important for the satellite cell niche. So we utilize the tissue clearing protocol established in our own laboratories and use the confocal 2-photon and light sheet microscopes. These are tibialis anterior muscle received injected cells inside. After tissue clearing, you can clearly see the LACZ positive cells inside in the deep inside of the skeletal muscle. To visualize satellite cells and the capillaries, we utilize the PAX7 Curi-ERT2 mediated TD tomato and Fruc1 GFP mice. You can see nice separation of these two populations by FACS and clear image for satellite cells and capillaries here. So now we can see deep tissue imaging for fluorescent reporters. Of course, red is the satellite cells and green is the capillaries. This is a whole soleus muscle satellite cell image. So then we use the light sheet microscope, which is faster and cheaper to capture big image. But there is some, background staining. But you can see all satellite cells in the big soleus, not big, but still the pretty much whole soleus muscle. Okay, interestingly, we noticed that satellite cells show multiple long projections like this. For example, this guy has many projections, look like an octopus tentacles. And these two satellite cells on the same fiber seems to be touching each other, look like a Michelangelo's painting. Okay, so this is another 3D image. So that we utilize the pseudo color for satellite cells. Initially, satellite cells are red, but then we utilize the different colors based on the proximity. So most of the satellite cells are pretty close to the capillaries, like here. Oops. However, for example, this guy, some satellite cells, like this orange cells, showed away from capillaries, like here. Okay. So this is a kind of the quantitative data. So that this histogram shows the distribution of distance of cells in extraocular muscle, EOM, soleus muscle, and also TA muscle. So most of the satellite cells show pretty close proximity to the capillaries, like here. So this is a distance. But some of them, especially TA muscle satellite cells, show away from capillaries. Based on the EDU labeling experiment, we noticed that satellite cells close to the capillaries are more stem-like populations. Okay, next question is how this proximity is established. To identify the signaling pathway involved in satellite cell capillary crosstalk by an unbiased approach, we utilized RNA-seq and interactome analysis, which means satellite cells express ligand and endothelial cells express the corresponding receptors. So among them, we noticed that VEGF pathway is the highest signaling pathway in this kind of screening. So among the VEGF family, we noticed that VEGF-A is highly expressed in both quiescent and also activated satellite cells on single muscle fibers. VEGF-A is also detected in myoblast, but expression is downregulated during differentiation. To show roles of satellite cell-derived VEGF-A, we utilize the PAX7 ERT2 mediated the VEGFA conditional knockout by flox mice. 3 weeks after knockout, which can be induced by the tamoxifen treatment, we saw the distance between satellite cells and the capillaries became wider, like this, indicating that satellite cell-derived VEGFA is essential for establishment of satellite cell and capillary proximity. So next, next question is how satellite cell self-renewal is regulated by vascular niche. Again, to find the potential signaling pathway from the endothelial cells to satellite cells, we did RNA-seq then interactome analysis. In that case, we screened the ligand should be expressed from endothelial cells, and receptors should be expressed in the satellite cells. By using this interactome analysis, we found that Notch pathway is the highest signaling pathway in this category. So as many groups already published that Notch ligand, which is usually JAG1, JAG2, delta-like 1, 3, and 4, activate Notch signaling in satellite cells, which can induce Notch effector gene expression like HES and HEY genes, which eventually suppresses MyoD activities and induces satellite cell self-renewal. So this way. However, it is still not so clear which Notch ligands are involved and which cell types produce the Notch ligands. And this is a published single-cell RNA-seq data in whole muscle. There are at least 8 different cell types based on the clustering. So, and then we check the Notch ligand expression in endothelial cells. And then we notice that the delta-like 4 is the highly expressed in the endothelial cells. So we focus on the endothelial cell-derived DLL4. Okay, so next, to analyze DLL4 function on satellite cells, we utilize the VE-cadherin-mediated endothelial cell specific DLL4 knockout mice. So the VE-cadherin ERT2 clearly marked GFP positive and lectin positive endothelial cells upon tamoxifen treatment. In endothelial cell specific DLL4 knockout mice, satellite cell number was significantly reduced only in injured skeletal muscle, but not a baseline, suggesting that endothelial cell derived DLL4 is essential for satellite cell self-renewal. So this is the current model. During muscle regeneration, satellite cell derived VEGFA recruits the capillaries to establish vascular niche for satellite cells. And then endothelial cell derived DLL4 induces Notch activation in neighboring satellite cells for self-renewal. So this is a working hypothesis. Vascular niche is important for satellite cell self-renewal and maintenance. In aging and dystrophic muscle, this proximity might be disrupted, and thus reversing from distant proximity to close proximity might be the potential therapeutic approach for these muscle defects. So Duchenne muscular dystrophy, or DMD, is an X-linked progressive muscle wasting disease, and patients die around the early 20s due to lack of functional dystrophin protein, which is localized to the inner face of the cell membrane, like here, and involved in mechanotransduction. So that you can see nice dystrophin protein expression in the muscle membrane, but it's gone in DMD patient. Consequently, muscle became very dystrophic, such as accumulation of the adipose and massive fibrosis and calcification. So we noticed that among VEGF signaling, VEGF-A is significantly reduced in human DMD patient, as well as the Becker-type muscular dystrophy patient. And also DMD dog and MDX mice, which is a DMD model, mouse models, indicating the importance of VEGF-A signaling in DMD. So there are two VEGF receptors, and FLUC1 is a major VEGF receptor and involved in cell migration, cell proliferation, migration, survival, and differentiation. By contrast, FLUC1, which is a high affinity VEGF receptor, but cytoplasmic signal is very weak, and thus works as a decoy receptor. And also there is a secreted FLUC1 protein called SFLT1, which also works as an inhibitor of the VEGF signaling via VEGF trap. So previously we showed that FLUC1 homozygous mice die early embryogenesis due to the excessive angiogenesis. So 10 years ago, we published that when FLUC1 heterozygous mice were crossed with MDX mice, vascular density was increased. Consequently, DMD-associated muscle pathology is improved, such as reduced calcification and reduced fibrosis. In addition, satellite cell number is increased. MDX utrophin double mutant mice are very severe DMD model mice, and they die within around 5 months, resembling human DMD pathology. When FLUC1 heterozygous mice were crossed with double mutant mice, the lifespan is significantly extended, indicating that increased vasculature might be beneficial for the DMDs. Okay, so then we have demonstrated that global FLUC1 het mice show the beneficial effect when crossed with MDX mice. After that publications, conditional FLUC1 KO mice became available and showed the similar increased vascular density phenotype when FLUC1 was postnatally deleted, like here. So to reveal which FLT1 deletion can show the beneficial effect on MDX mice, we generated 3 different conditional FLT1 knockout mice. One is a postnatal global conditional knockout, and the second is a postnatal satellite cell specific knockout, and postnatal endothelial cell specific knockout. As a result, endothelial cell specific FLT1 knockout mice could recapitulate the improved DMD phenotype shown in the original FLT1 het mice. Okay, so therefore we thought postnatal FLT1 blocking might be a novel therapeutic approach for DMD. By collaboration with Shire, now part of Takeda Pharmaceuticals, we developed monoclonal anti-FLT1 antibodies, which can block VEGF-FLT1 binding. Normally, VEGF is trapped by FLT1, which is a high affinity decoy receptor, limiting FLT1 cytoplasmic activation. So that since FLT1 is the main kind of the functional VEGF receptor. So treatment with anti-FLT1 antibodies, which blocks VEGF-FLT1 binding, increases available VEGF pool, and activate FLT1 more. This strategy might be beneficial for the DMD. So after intravenous injection of anti-FLT1 monoclonal antibodies into MDX mice, so 1 month later, we saw increased serum VEGF level and also decreased soluble FLT1 levels. Consequently, we saw increased vascular density and reduced Evans blue dye positive injured skeletal muscle and reduced fibrosis. And finally, we saw increased grip strength and also free running capabilities. Finally, we also saw the increased satellite cell population. So taken together, these data indicate that the anti-FLT1 antibody is a novel therapeutic drug for the DMD. Okay, so while endothelial cell specific FLT1 knockout mice show the beneficial effect when crossed with the MDX mice, like here, global FLT1 knockout mice show the deleterious phenotypes. Why do global FLT1 knockout mice display deleterious muscle phenotype in MDX mice? So this is a question. Previously, we showed that mildly knockout myoblasts show a signature of endothelial cell gene expression, such as expression of the SCA1, CD34, and also VCAM1. Developmentally, somite-derived myoangioblast, which express both FLT1 and PAX3. FLT1 is, of course, the endothelial cell marker, and the PAX3 is a myogenic marker, migrate into limbad to give rise to both myogenic and angiogenic cells. Therefore, we may be able to find the endothelial cell signature in adult satellite cells. To find this answer, we performed RNA-seq analysis for the muscle-derived endothelial cells, satellite cells, and also muscle fibers. So we noticed that satellite cells express some canonical endothelial cell markers, such as CDH5, which is V cadherin, yes, and PCAM1, and also FRUT1. Like here. Okay, so 20 years ago, Julio Kosu reported that aorta-derived mesoangioblast and satellite cells display similar transcriptional profiles, such as the expression of the both myogenic and endothelial cell markers. More recently, whole adult muscle single-cell proteomics showed that satellite cells express endothelial cell markers, such as a PCAM1, with a lower amount. And also RNA scope also support this idea that V cadherin message is detectable on in the satellite cells. To confirm this, we performed single-cell RNA-seq for the isolated satellite cells and endothelial cells from both intact muscle and CTX injured muscle at day 3. So essentially, we did a deep sequencing with 300K reads per cell basis, compared with normally 60K reads per cell, to identify minor gene expression in each cell population. The aggregated data clearly showed the quiescent satellite cells, activated satellite cells, and myblast population, and also endothelial cell population, like here. And interestingly, we noticed that FLUC1 GFP and V cadherin are clearly expressed in some satellite cells. In addition, VEGF receptors FLUC1, FLUT1, and also co-receptors NRP1 and NRP2 are also expressed in the at least 20 to 30% of satellite cells. We also confirmed endothelial cell expression, including FLUT1, by using RT-PCR, immunostaining, and also the RNA scope. While FLUC1 expression in satellite cells is 100 times lower than the endothelial cells, FLUT1 expression in satellite cells is relatively high. Immunostaining also confirmed low level of FLT1, but not FLUC1 in satellite cells. And the RNA scope also confirmed the FLUT1 message in satellite cells. Then we examined whether VEGF FLUT1 signaling is important for the satellite cell function. We first examined the cell proliferation and differentiation assay. However, there is no obvious difference after treatment of the VEGF for the proliferation and the differentiation. Next, we induced cell apoptosis after treatment with tapsgalgin, which can induce ER stress-mediated apoptosis. Interestingly, apoptotic cells are reduced when VEGF was treated. but apoptosis is increased. This is a control. Apoptosis is increased when the FRUT1 gene was deleted. So that we isolated the satellite cells, derived myoblasts from satellite cell specific FRUT1 knockout mice. So that after multiple experiment, which I didn't show here, but we conclude that so the VEGF-A, FRUT1, and AKT phosphorylation signaling is important for the satellite cell survival in vitro. So there are hypomorphic VEGF-A transgenic mice termed VEGF-A-Hyper mice, in which VEGF-A expression levels are 1.5-fold upregulated. So we crossed these VEGF-A-Hyper mice with a PAC7 TD tomato mice and injected tamoxifen to induce TD tomato in satellite cells, and we isolated the single muscle fibers, or we injected CTX to induce muscle regeneration. As a result, satellite cell death via necrosis is significantly reduced in the VEGF-A-Hyper mice and increase the satellite cell number after regeneration. Consequently, muscle regeneration shows faster compared to the wild-type mice. So next, we established the satellite cell-specific VEGF-A knockout mice by crossing PAC7 CRE-ERT2 with VEGF-A floxed mice. So VEGF-A conditional knockout mice clearly showed the increased number of apoptosis during muscle regeneration. And consequently, satellite cell number was decreased and muscle fiber diameter became smaller after injury. Interestingly, oil red positive adipose was also increased in satellite cell specific VEGF-A knockout mice. Therefore, satellite cells derived VEGF-A is essential for proper muscle regeneration. Okay, lastly, we established the satellite cell specific FRUT1 knockout mice in which PAC7 CRE-ERT2 mice were crossed with FRUT1 floxed mice. They clearly display increased cell apoptosis during muscle regeneration, and consequently, satellite cell number was decreased and muscle fiber diameter became smaller after injury. Therefore, VEGF-A FRUT1 signaling in satellite cells is essential for proper muscle regeneration. Okay, so this is a current working model. Satellite cell derived VEGF-A is essential for cell survival and self-renewal during muscle regeneration, which is mediated by the FRUT1 as a VEGF-A receptor. So that means the FRUT1 function is dependent on the cell types. For example, this case works as the satellite cell case works as a positive receptor because the FRUT1 expression is absent. So the FRUT1 is the only receptor for VEGF. By contrast, in the endothelial cells, FRUT1 works as a negative receptor because the FRUT1 become a decoy receptor, but the FRUT1 works as a major receptor for the signaling. Okay, so the ETV2 is zinc finger proteins, transcription factors, works as an endothelial cell reprogramming factors. Overexpression of ETV2 convert human dermal fibroblast into endothelial cells. So that since satellite cells show some endothelial cell signature, we examined whether overexpression of ETV2 can convert satellite cells into endothelial cells. So this is a result. So satellite cells are usually round shape and they do not express any FLUC1 GFP when isolated from FLUC1 GFP mice. After ETV2 induction, they became a cobblestone appearance like here, and FLUC1 GFP become positive. And these cells also express FLUC1 proteins and also VE cadhein protein around the tight junctions area, indicating the successful endothelial cell conversion of muscle satellite cells. Okay, so then for the future directions, we are addressing following questions. What is the functional differences between endothelial cell marker positive and negative satellite cells? And also, are satellite cells derived from endothelial cell marker positive myoangioblast or not? And finally, how can ETV2 reprogram MyoD positive myogenic cells to endothelial cell lineages? So cell lineage tracing and epigenetic experiment will answer these questions. Okay, so for the last part, I would like to show circadian rhythms and muscle regeneration. So circadian rhythms react to light from the outside world. The suprachlasmatic nucleus is in the hypothalamus, is the center of circadian rhythms. And circadian rhythm exists in the whole body's organ, including skeletal muscle. They are regulated by the secreted factors such as glucocorticoid, melatonin, and testosterone. RNA-seq data revealed that around 10% of the genes in each tissue show some circadian expression manner, like here. Okay, the molecular function of circadian rhythm is conserved from fly to mammalian. Both positive circadian transcription factors such as clock and BMAL1 and negative factors PAR and CRY regulate 24 hours circadian rhythms by a feedback loop. It is well known that muscle function and metabolism are regulated by MyoD and PGC1 beta as a master transcription factor. BMAL1 and clock control circadian expression of both MyoD and PGC1 beta. And as a negative factor, RB and also PAR1, PAR2, CRY1, CRY2 works also involved in the circadian regulations as a negative factor. Okay, so the circadian rhythms are also important for the satellite cells in aged muscle, in which circadian rhythms of satellite cells rewire to adapt to metabolic cues and also to age-related traits, so that they have a still circadian rhythm, but the gene expression patterns are kind of rewired. This rewiring is prevented by the caloric restriction in aged mice. So, and also there is a report that mouse dermal fibroblast migration is faster in night compared to daytime due to the difference of actin microfilament organization. In human, there is a clinical evidence that skin burned in daytime heals much faster than skin burned in nighttime. So therefore, we questioned whether there is any difference in muscle regeneration between daytime and nighttime induced injury. So we inject the beryllium chloride to induce muscle injury at 9 a.m. or 9 p.m. And exactly 4.5 days later, we harvest the muscle. So interestingly, muscle regeneration is slightly faster when 9 p.m. injection of beryllium chloride compared to the 9 a.m. injection. This difference is conserved in both male and female mice. In addition, by day 14, Sirius red positive fibrosis is more evident in the 9 a.m. injured muscle compared to more evident in 9 a.m. compared to 9 p.m. injured muscle, indicating that there is a long-term effect of circadian rhythm dependent muscle regeneration. Okay, so it is reported that Per1 or Per2 knockout mice, both are the important clock genes, display blunted circadian rhythms. Therefore, we examined muscle regeneration capacity in Per1, Per2, or Per1, Per2 double knockout mice. Clearly, muscle regeneration is delayed in Per1 or Per2 mice, and more drastically in double knockout mice, indicating the essential roles of Per1 and Per2 genes in muscle regeneration. So as I showed that wild type mice show the faster muscle regeneration when the muscle injury was initiated at in PM compared to AM, like here. However, this difference disappeared by Per1 knockout and Per2 knockout, and also double knockout. This tendency is also repeated when we utilize the embryonic myosin heavy chain staining, which detect newly formed regenerating muscle fibers. Okay, so now we shift the in vitro assay. When Per1 or Per2 genes were knocked down by shRNA or knockout by CRISPR-Cas9, myosin heavy chain positive myogenic differentiation is markedly reduced in C2-C12 myoblast. Muscle fusion index is also reduced when Per1 or Per2 genes were knocked down or knockout, indicating that Per1 and Per2 is essential for the normal myogenic differentiation in vitro. So we performed RNA-seq analysis in wild type, Per1 and Per2 knockout myoblast. And then we noticed that decrease muscle specific gene expression in PAR1 and PAR2 knockout cells compared to control cells. Among them, we picked up insulin-like growth factor 2 or IGF-2. Since IGF-2 is known to positively regulate myogenic differentiation. As you can see, IGF-2 expression is significantly reduced in PAR1, PAR2, knockdown, and knockout cells, indicating that IGF-2 is a PAR1, PAR2 downstream gene. Next, we performed IGF-2 gene knockdown in C2C12 cells and showed that myogenic differentiation is significantly reduced in IGF-2 knockdown cells compared to the control cells, like here. To see whether myoblast possess cell autonomous circadian clock, we isolated the satellite cells from Per2 luciferase transgenic mice. And then dexamethasone was used for synchronization of circadian clock. And luminometer culture system was performed. Clearly, we can detect the nice 24 hours circadian oscillation of luciferase activities, at least for 4 days, indicating that satellite cell-derived myoblasts maintain cell autonomous circadian clock after isolation. So we utilize the C2C12 cells, which also showed the nice 24 hours oscillation after synchronization by dexamethasone. So after synchronization, we harvested the cells every 4 hours. And then we performed Western blotting. And then we show that BMAR1, PAR1, PAR2, IGF-2, and IGF-2 downstream proteins, phosphor P38, showed a very nice 24 hours oscillation. BMAR1 expression showed that different timing compared with PAR1, PAR2, and IGF-2, and phosphor P38. Interestingly, PAR1 or PAR2 knockout cells display significantly reduced expression of the IGF-2. So you can see IGF-2 is gone, and also phosphor P38 is also gone, indicating that IGF-2 is the downstream effector protein of PAR1 and PAR2. So IGF-2 enhancer is located near NCTC1 gene downstream of IGF-2. Epigenetic analysis show that there are peaks of BMAR1, histone H3K27 acetylation, histone H3K4 monomethylation, and also P300, which is histone deacetylase. And also these histone marks as a mark for enhancer region. So interestingly, these peaks also show oscillated manner like this, 24 hours oscillated manners. When PAR1 or PAR2 were knocked out, circadian peaks of H3K27 and H3K4 monomethylation were disappeared, indicating that PAR1 and PAR2 mediated epigenetic enhancer modifications regulate circadian IGF-2 expressions. Okay, so finally, we utilized PAC7 CRI-ERT2 IGF-2 LOXP mice for the satellite cell specific IGF-2 knockout mice to see if IGF-2 is involved in the muscle regeneration. So we injected a tamoxifen to delete the IGF-2 gene in satellite cells, and then beryllium chloride was injected into muscle to induce a muscle regeneration. Clearly, we see reduced muscle fiber diameters in both intact IGF-2 knockout and also more significantly reduced muscle regeneration in IGF-2 knockout mice compared with control wild type mice, indicating the essential roles of IGF-2 in proper muscle growth as well as regeneration. Okay, so that this is current working model. Per1 and Per2 mediated IGF-2 expression plays an essential role in myogenic differentiation. Per1, Per2, and IGF-2 expression is higher in the evening compared to morning time, which promote myogenic differentiation more efficiently in the evening time compared to the morning time. Okay, so I would like to thank our collaborators, especially the Nobuaki Kikyo, who is a collaborator for the circadian rhythm experiment in University of Minnesota, and Dr. Kraus for the RNA scope, Dr. Kubota for the V cadherin Cray ERT2 mice, and Dr. Ferrara for the VEGFA mice, and Dr. Shima for the DL4 mice, and Dr. Ima for the FLUC1 GFP mice. I also want to thank for our laboratories members, especially former student Mayank Burma, and also Yuko Shimizu Motohashi for the contributions. Thank you very much.
Thank you very much as well, Atsushi. What a wonderful seminar. Thank you.
Thank you.
Very clear, beautiful slides, I've got to say. So I'd ask people to please go to the Q&A box to ask your questions.